Hi everyone, it's Dr. Fox, and let's talk about renal regulation. In this video, we'll discuss intrinsic and extrinsic control of renal function, we'll outline the RAA system, and we'll map the autonomic innervation of the kidneys. So let's begin with renal regulation. The most significant control of the nephrons is intrinsic versus extrinsic control. The two methods of intrinsic control are the myogenic response, which is basically just the contraction of smooth muscle within the afferent arteriole when they're stretched, and tubulo feedback, meaning whenever specialized chemoreceptors within the tubules of the nephrons sense too high or too low a concentration of sodium chloride, they can adjust accordingly. The remainder of control is extrinsic to the kidneys, and this comes from the autonomic nervous system, specifically the renal plexus. The renal plexus is an autonomic plexus which is located on the renal arteries. The important component of the autonomic response from the renal plexus is sympathetic stimulation of or from the renal plexus causes vasoconstriction, specifically vasoconstriction of the afferent arterioles. Vasoconstriction of the afferent arterioles is going to decrease glomerular filtration rate. So it is going to maintain more blood within the cardiovascular system, and thus less filtrate moves in through the urinary system. The other means of extrinsic control is that of hormones, specifically through the renin-angiotensin-aldosterone system, or the RAA pathway, and through antidiuretic hormone, also known as vasopressin. These systems of hormones are going to increase water reabsorption from the filtrate back into the cardiovascular system. Now, there are other hormones, so atrial natriuretic peptide produced by the heart and brain natriuretic peptide, which are going to oppose that and cause an increase in water excretion. So not all hormones are going to conserve water, some will excrete it. So let's take a look at the general vascular response within kidneys and see how that is going to affect glomerular filtration rate, or GFR. Now, the two major sites of action are going to be the afferent arteriole and the efferent arteriole. So the afferent arteriole is the primary site of action in terms of vascular response. Its diameter is going to be proportional to glomerular filtration rate, meaning whenever a stimulus is going to cause vasoconstriction or the narrowing of the afferent arteriole, glomerular filtration rate will decrease proportionately. Whenever there is vasodilation or relaxation of that smooth muscle, glomerular filtration rate will increase proportionately. Now, it is exactly the opposite with the afferent arteriole, where the diameter of the efferent arteriole is inversely proportional to glomerular filtration rate, such that whenever we vasoconstrict, whenever we slow down blood leaving the glomerulus, that's going to increase glomerular filtration rate. And whenever we vasodilate, that is, whenever we increase the flow of blood outside of the glomerulus, that's going to decrease glomerular filtration rate. So think of these things as supply. If there is more supply going into the glomerulus, glomerular filtration rate increases, less it decreases. Whenever that supply is held within the glomerulus for longer through vasoconstriction of that efferent arteriole, glomerular filtration rate will increase and inversely decrease when it is vasodilated and blood leaves. So let's take a look at afferent arteriole autoregulation. So autoregulation, or intrinsic control, is the most significant, and there are two major mechanisms. There's myogenic regulation as well as tubuloglomerular feedback. So within the afferent arteriole, we have smooth muscles within the walls. It is baroreceptive, meaning it can sense if it is being stretched or not. And with stretching, if there is an increase in blood pressure, that is going to cause vasoconstriction. Vasoconstriction is going to lower glomerular filtration rate. Now, on the opposite side of the coin, if those baroreceptors of the smooth muscle sense a relaxation, that's going to cause a vasodilation. With vasodilation of the afferent arteriole, we're going to get an increase in glomerular filtration rate. So this is the first line and most significant line of autoregulation within the kidneys. The second line is going to come from the juxtaglomerular apparatus. So juxtaglomerular, juxta means next to, near, or adjacent. Glomerular, the glomerulus, or the JG apparatus, is a collection of tissues that is just adjacent to the glomerulus and the renal corpuscle writ large. So this is nestled between and of the distal convoluted tubule and the afferent and efferent arterioles. So let's take a look at the components. The first is the macula densa. The macula densa is a specialized group of cells within the distal convoluted tubule, and what it does is it responds to concentrations of salt. So it's a chemoreceptor, and with high salt concentrations, it causes the afferent arterioles to constrict. So these will narrow as a result. So we're using salt as a proxy for glomerular filtration rate, because the higher the amount of concentration of salt, the higher the glomerular filtration rate. The lower the concentration of salt, the lower the glomerular filtration rate. So with high salt concentrations, we have vasoconstriction of that afferent arteriole. Therefore, we have a reduction in GFR, glomerular filtration rate. The other side of things, if we have a low concentration of salt, that causes the afferent arterioles to vasodilate. They get larger, and so this is going to lead to an increase in glomerular filtration rate. There are also specialized juxtaglomerular cells, and these are going to be in the periphery here, and these will also be baroreceptive for the afferent arterioles, in addition to the baroreceptive ability of the smooth muscle within the walls. But these are more significant in terms of their role that they play in the RAA system or pathway, in that they release a hormone known as renin. So let's take a look at the RAA system. RAA stands for renin-angiotensin aldosterone. This is a system which consists of a series of catalyzed reactions that come in response to a drop in blood pressure. Now, the major triggers for the activation of the RAA system are low blood pressure, a detection of low salt in the distal convoluted tubule by the macula densa, or direct sympathetic stimulation. The first response to these triggers is the secretion of renin by the juxtaglomerular cells of the kidneys. Renin is an enzyme, and it converts a precursor called angiotensinogen, which is secreted by the liver. And it converts angiotensinogen into angiotensin 1. Angiotensin 1 can then be converted into angiotensin 2 by angiotensin converting enzyme. Angiotensin converting enzyme is oftentimes referred to as ACE. So you may have heard of ACE inhibitors. They inhibit the release and action of ACE. So this reaction, angiotensin 1 to angiotensin 2 by ACE, occurs on the lining of the pulmonary endothelia. Angiotensin 2 is capable of a number of effects. It is a potent vasoconstrictor. So when we think of a response to a decrease in blood pressure, vasoconstriction is going to be an immediate opposition to that. Through vasoconstriction, we can elevate blood pressure. It is a particularly potent vasoconstrictor on the efferent arteriole. And this is important for the function of the kidney because even though this is going to increase glomerular filtration rate, there's going to also be concurrent vasoconstriction of the afferent arteriole. And so this balances out that effect, and it allows glomerular filtration to continue during this process. Now, angiotensin 2 also inspires the adrenal cortex to release aldosterone. So the suprarenal gland is completing the RAA. That ultimate A is for aldosterone. And aldosterone encourages the movement of sodium and then water from the filtrate of the distal convoluted tubule back into the peritubular capillaries. So aldosterone is encouraging reabsorption in the distal convoluted tubule of sodium and water. Angiotensin 2 also stimulates the hypothalamus to release a special hormone called ADH, which stands for antidiuretic hormone. You may have heard of ADH by its more common name, vasopressin. Vasopressor. Diuretic hormone, or vasopressin, accomplishes two things. One, much like aldosterone, it encourages sodium and water reabsorption in the distal convoluted tubules. So we're maintaining our water within the body, not releasing it as urine. Secondly, and this is in the name, vasopressin is a powerful vasoconstrictor. So we have all of these substances which are opposing the drop of blood pressure. Now let's take a look at the renal plexus, but let's start more generally with the autonomics of the abdominal cavity. So the abdominal cavity has a series of autonomic ganglia and plexuses, and all of these are associated with and named for vessels that they are in close proximity to. So for instance, there are going to be celiac ganglia and plexuses, superior mesenteric ganglia and plexuses, inferior mesenteric ganglia and plexuses, renal ganglia, and so on and so forth. These get their inputs through two major sources. So sympathetic chains, these are the paravertebral ganglia. The sympathetic chains get all of their input from spinal levels T1 through L2. So the spine provides input to the sympathetic chains at the levels of T1 through L2 of those spinal nerves. There are also splanchnic nerves from both the thoracic chains, these are thoracic splanchnic nerves, as well as the lumbar chains, those are lumbar splanchnic nerves. The other major source comes from the posterior vagal trunk. The anterior vagal trunk helps a little bit with the foregut, so stomach, liver, and gallbladder, but it's the posterior vagal trunk that provides the parasympathetic inputs to these pre-aortic ganglia and plexuses. And as we've named, they are associated with major vessels. So celiac ganglion plexus, superior mesenteric ganglion plexus. There's also an aortico-renal that is adjacent to both the aorta and the renal vessels. Renal plexuses, which are on the renal arteries, and small ganglia located posterior to that, as well as an intermesenteric plexus that's between the SMA and IMA upon the aorta. So let's look at this specifically for the renal plexus. There are two renal plexuses, and these are associated with each renal artery. There are also small ganglia posterior to the renal arteries near their origins. Predominantly, the effect from this plexus is going to be a sympathetic response. So with this, we get vasoconstriction, especially vasoconstriction of the afferent arterioles, and we get the secretion of renin from the juxtaglomerular, or JG, cells. The major input to the renal plexus comes from the celiac ganglia and plexus, as well as aortorenal ganglia and plexus. These are coming from the thoracic spine, as well as the lumbar spine. Thoracic splanchnic nerves in particular. So there's a greater, a lesser, and a least thoracic splanchnic nerve. When it's present, it's the least thoracic splanchnic nerve that innervates this renal plexus directly. We also get input from the first lumbar splanchnic nerve, as well as some from the intermesenteric plexus. So there are a variety of autonomic sources, but predominantly these are coming from the thoracic spine, in particular the least thoracic splanchnic nerve when it is present, as well as the first lumbar splanchnic nerve. So let's assess the knowledge. When present, which thoracic splanchnic nerve provides direct sympathetic innervation to the renal plexus? Is it the first lumbar splanchnic? Nope, that's not a thoracic splanchnic. Second, nope, that's not it either. Greater thoracic splanchnic nerve, that will provide innervation by means of the celiac and SMA, so there's some there. The least, yes, that is the correct answer. That's what directly innervates it. And the lesser, yes, you get some of that through the celiac and aortorenal plexuses, but least thoracic splanchnic nerve is the best answer here. Thank you so much for your time, and I look forward to talking to you in the next video. Bye.